Mitochondria are the sites of cellular respiration, the metabolic process that uses oxygen to drive the generation of ATP by extracting energy from sugars, fats, and other fuels. The mitochondria are oval-shaped organelles found in most eukaryotic cells. They are considered to be the powerhouses of the cell. As the site of cellular respiration, mitochondria serve to transform molecules such as glucose into an energy molecule known as adenosine triphosphate. ATP fuels cellular processes by breaking its high-energy chemical bonds. Mitochondria are most plentiful in cells that require significant amounts of energy to function, such as liver and muscle cells. Mitochondria produce energy in the form of adenosine triphosphate or ATP. It is the major energy currency of the cell that provides the energy for most of the energy-consuming activities of the cell. The ATP regulates many biochemical pathways. Here's the mechanism. When the third phosphate group of ATP is removed by hydrolysis, a substantial amount of free energy is released. In animals, ATP is produced in the mitochondria through the process called cellular respiration. In plants, it occurs in the chloroplast by the process called photosynthesis. Most energy-consuming activities of cells, such as joining transfer RNAs to amino acids for assembly into proteins, synthesis of nucleoside triphosphates for assembly into DNA and RNA, synthesis of polysaccharides, synthesis of fats, active transport of molecules and ions, conduction of nerve impulses, maintenance of cell volume by osmosis, addition of phosphate groups to different proteins, beating of cilia and flagella, including sperm, and bioluminescence. In mammals, ATP also functions outside of cells. ATP is released from damaged cells to elicit inflammation and pain. It is released from the carotid body to signal a shortage of oxygen in the blood. ATP is released from taste receptor cells to trigger action potentials in the sensory nerves leading back to the brain, and it is also released from the stretched wall of the urinary bladder to signal when the bladder needs emptying. In eukaryotic cells, the mitochondria and chloroplasts are the organelles that convert energy to other forms which cells can use for their functions. Chloroplasts, which are found in plants and algae, are the sites of photosynthesis. The word chloroplast is derived from the Greek word chloros, which means green, and plasts, which means the one who forms. The chloroplasts are cellular organelles of green plants and some eukaryotic organisms. These organelles conduct photosynthesis. They absorb sunlight and convert it into sugar molecules. They also produce free energy stored in the form of ATP and NADPH through photosynthesis. Chloroplasts are double membrane-bound organelles and are the sites of photosynthesis. The chloroplast has a system of 3 membranes: the outer membrane, the inner membrane, and the thylakoid system. The outer membrane is a semiporous membrane and is permeable to small molecules and ions which diffuse easily. The outer membrane is not permeable to larger proteins. The inner membrane of the chloroplast forms a border to the stroma. It regulates passage of materials in and out of the chloroplast. In addition to the regulation activity, fatty acids, lipids, and carotenoids are synthesized in the inner chloroplast membrane. The stroma makes up much of the volume of the chloroplast. Stromathiss is an alkaline, aqueous fluid that is protein-rich and is present within the inner membrane of the chloroplast. It is the space outside the thylakoid space. The chloroplast DNA, chloroplast ribosomes, thylakoid system, starch granules, and other proteins are found floating around the stroma. The thylakoid system floats in the stroma. The word thylakoid is derived from the Greek word thylakos, which means sac. Thylakoids are small sacs that are interconnected. The membranes of these thylakoids are the sites for the light reactions of the photosynthesis to take place. The chlorophyll is found in the thylakoids.
The thylakoids are arranged in stacks known as grana. Each granum contains around 10 to 20 thylakoid.
The molecules present in the thylakoid membrane use the electrons that are energized to pump hydrogen ions into the thylakoid space. This decreases the pH and causes it to become acidic in nature. Thylakoids are of two types, granal thylakoids and stromal thylakoids. Granal thylakoids are arranged in the grana. These circular discs that are about 300 to 600 nanometers in diameter. The stromal thylakoids are in contact with the stroma and are in the form of helicoid sheets. The granal thylakoids contain only photosystem II protein complex. This allows them to stack tightly and form many granal layers with granal membrane. This structure increases stability and surface area for the capture of light.